Hi, we're Team iGEM Laval, and we want you to picture this. It's springtime in Eastern Canada, and nights are still freezing, way below 0 degrees Celsius, but days can get pretty warm, climbing well above the freezing point. Let's take a look at what happens to a drop of sap from a sweet sugar maple tree during this season. During the cold night, the sap goes up into the xylem of the tree, but during the warmer day, it makes its way back down to the roots. Some people know this process very well. By piercing the sugar maple's trunk, they harvest the sap of the tree, collecting it in a tank with the help of a tubing system. Once the sap is in that tank, it's officially on its way to becoming something great. The already slightly sweet sap is first concentrated through reversed osmosis. Then, after evaporating the water that's left in the sap, after that, we get maple syrup. It has a big place in Canadian people's hearts and recipes. It's also got a pretty decent spot in Canada's economy. According to Statistics Canada, in 2021 alone, Canada produced over 558 million Canadian dollars worth of maple syrup products. That's over 441 million US dollars. Although some bacteria in the maple syrup create desirable chemicals like vanillin and simple sugars, some bacteria can create excessive amounts of dextrins, long, complex sugar molecules. They cause problems like ropey maple syrup. Luckily, aSAP's here to help. We're making a treatment to avoid wasting this defective syrup. Specifically, we're making a dextrinase, an enzymatic treatment that uses enzymes to cut these long dextrins into smaller pieces, making the syrup's texture more manageable.
So to accomplish our project, we have set ourselves a couple of goals throughout the 2 years we have been working on the project aSAP. In 2020, since we didn't have any access to a lab, we focused on the design of our project by contacting maple producers and experts in the field to have a better understanding of the industry and the problematic of ropey maple syrup. Based on their feedback, we searched for potential dextrinases, from which we selected the dextrinase of JD-Bacter allergens and designed the plasmid for its expression with its sequence modified based on a previous study. And in parallel, we simulated the dynamics of the protein in function of the temperature. For this year, we focused our efforts on the implementation of our project by making the proof of concept with enzymology and rheology analysis, modeling the mixability of the enzyme in ropey maple syrup, analyzing the microbiology, and continuing working hand in hand with legal and regulative entities.
This led to some pretty solidly integrated human practices. As we said while talking about our project goals, last year we mostly talked to experts and stakeholders to know where our project was going. This year, though, we focused on a more practical approach. How can we help stakeholders turn aSAP from a research project into a usable solution? First, how will products created with aSAP be seen by consumers? We conducted a consumer survey, collecting answers from more than 300 people. From these answers, we learned that consumers see value in projects like ours, but they don't really understand much about enzymatic treatments in food. Next, how do we make sure that aSAP fits into the legal context of the field we're making it for? We had a second meeting with the MAPAC, the government agency that's in charge of most food regulations in Quebec, the Canadian province our team is based in. They gave us more information about the legal context in which we could implement aSAP. They were also curious about our project and were open to the idea of new regulations regarding the maple industry, since those haven't changed in the last 40 years. Finally, how are we going to make it easier for our project stakeholders to continue with our project after iGEM? We had more meetings with the PPAQ, the biggest maple syrup producers association in Quebec. They gave us general feedback on where our project could go to make things more interesting, and they gave us ropey syrup samples to help us succeed in our proof of concept and microbiology. Their key insight is also what brought us to on our path to modeling mixing processes.
Now that we have exposed our goals and the ways that our integrated human practices impacted our project, it's time to dive into the methodology. As mentioned before, the main goal of our project is to use Gilidibacter algens dextranase to degrade dextrans in ropey maple syrup and thus reduce its viscosity. The three main steps to achieve the proof of concept is 1. Express the protein in a bacterial system and purify it using iMac chromatography. 2. Assess the activity of the dextranase using Nelson-Samoyi assays in controlled conditions and then try it in the ropey maple syrup. And finally, quantify the viscosity of the treated ropey maple syrup with a rheometer. A rheometer allows to assess the physical properties of a liquid, such as its viscosity. In our case, we want to assess the decrease in viscosity of the treated ropey maple syrup versus a control. In parallel to our proof of concept, we did simulations to find out which mixing strategy would be the best to implement our treatment into the ropey maple syrup barrel. Also, as an attempt to excel in another area, we did the microbiological characterizations of 3 ropey maple syrup samples. The first step of our experiments started in 2020 when we designed the expression vector we use this year. The PET28A vector allows the overexpression of the gene clone, and it is under the control of the T7 promoter. And when EPTG is added, the overexpression starts. Histidine tag is added in 3 terminal of the coding sequence, and this tag is used to purify the protein by immobilized metal ion affinity chromatography later on. And as mentioned earlier, the sequence clone is engineered based on our previous study and is codon optimized for the expression in Escherichia coli. So the methodology presented led us to produce the results demonstrating the proof of concept of aSAP. The first experiment we did was to determine which condition was the best to express the dextranase of Gilidibacter algens in Escherichia coli system. So we tested multiple conditions of EPTG concentration, temperature, and time of incubations. And the dextranase has a molecular weight of 64 kilodaltons. So we indeed observed the expression of this at this weight for many conditions. However, we observed the higher amount of protein after 18 hours of induction at 18 Celsius and at the concentrations of 0.4 millimolar and 1 millimolar EPTG. So now that we have successfully expressed our protein, the next question was, is the dextranase soluble? To answer this question, we performed the sonication of the sample at 1 millimolar and 0.4 millimolar EPTG. And after separating the soluble fraction and the insoluble proteins by centrifugation, we put the soluble fraction on STS page. We observe indeed that the dextranase is soluble and that the conditions that provide the higher amount of soluble proteins is 1 millimolar EPTG. So we move forward with the purification of this sample. Using iMac, we perform the purification and put the purification samples on SDS page. We observe that an important amount of dextranase is lost during the purification process. However, still a high amount of protein is collected in the elution. We see that the protein purified is highly pure, which is what we needed to pursue the experiments. It was also the first time that anyone successfully expressed and purified the Gilidibacter algens dextranase. So now that we have our pure dextranase, the remaining question was, is our dextranase active in the ropey maple syrup conditions? And this question led us to the enzymatic assay results.
Having preliminarily verified that our enzyme was indeed active, we tested the effect of pH and temperature on the activity and the stability of the dextranase using an adapted protocol of Nielsen-Somogyi assay. With the amount of released glucose equivalents by the degradation of dextran T40, we found that our enzyme is weakly affected by the pH tested, whereas it was more affected by the temperature. Indeed, the stability is higher at temperature below 20 degrees, while the activity is higher at 40 degrees. It is to note that at temperature between 15 and 20 degrees, the enzyme is still very active. Since the desired conditions of the treatment are at pH between 5.5 and 6.5, and at temperature between 15 degrees and 20 degrees, the dextranase of iGEMs would be well suited to the conditions of rupee maple syrup. This brings us to the question, will we be able to reduce the viscosity of rupee syrup as a real treatment? Here, we visually tested the effect of the dextranase on rupee syrup, and as you can see, the strings that are in the sample without the dextranase on your left are not present in the one with the dextranase, which indicates qualitatively it is indeed active in rupee syrup and it's able to reduce its viscosity. In order to have a quantitative data of the reduction, we proceeded with rheological assays in which we incubated rupee syrup with the dextranase at room temperature for 25 minutes before the measurement. With measurement of both our treated sample and a control sample at 10 degrees, to have a better view of the difference in viscosity, we can see that the treated sample with our dextranase has indeed a reduced viscosity compared to the control sample. This represents the proof of concept of our project.
Following last year's modeling that led to the experimental confirmation and characterization of our chosen enzyme, this year's modeling focused on the implementation of aSAP. To establish how aSAP would work in a real-world scenario, we wanted to find a way to mix our enzyme with rupee maple syrup. Through our human practices, we learned about the current processing and management of maple syrup from some of the industry's organizations like the PPAQ. To make implementation easier for our project, we didn't really want to make big changes in the way rupee maple syrup is being processed compared to regular maple syrup. So we took the existing steps in the process and built our implementation around it. When regular maple syrup is produced, maple producers store it in barrels. These barrels are sent to the PPAQ, who perform quality testing before storing them in warehouses. We want to implement our enzymatic treatment after rupee syrup fails these quality tests. The enzyme will be mixed into the rupee maple syrup with a mixer, and then treated rupee syrup will be stored in warehouses instead of being destroyed. How we will mix it though? That's where our modeling comes in play. To simulate the mixing process, we use two-dimensional computational fluid dynamics. Looking into mixing processes with the help of a chemical engineering expert, we found two types of model that could work for our intended purposes. Both worked in tanks that could contain 1 barrel worth of rupee syrup. The first one is a jet mixers that create a mixing flow by pushing fluid out of a nozzle at the bottom of the tank. We simulated the enzyme and rupee syrup coming out of the nozzle to study how well the enzyme could be mixed in using this model. We can see that the rupee syrup in red does not mix completely with the enzyme in blue at the end of the simulation. We later realized that the jet mixer will not be feasible in a real-world scenario because rupee maple syrup is too viscous. Indeed, that would make it hard for it to be pushed out of the nozzle. We then simulated our second model, a paddle mixer. In this model, the enzyme would be injected through the sides of the tank and be mixed by 3 rotating paddles. In the simulation, we can see that almost all the rupee syrup in red has been mixed with the enzyme in blue. Between the two models simulated, we can conclude that the paddle mixer would be the best. It also does not suffer from the viscosity of ropey syrup as much as the jet mixer.
Over the past 2 years, we realized how under-researched ropey maple syrup is. We wanted to help researchers gain more knowledge in this field by going above and beyond what we're doing with aSAP, our main project. To do so, we wanted to identify microorganisms present in ropey syrup because to date, there's only one paper that has done it. We hope to find new microorganisms, determine their origin, and help researchers manage this problem by identifying those that produce dextrins and determine which ones could originate from a human contamination. To briefly describe how we identified the microorganisms, we started with 3 samples of ropey maple syrup, from which we isolated colonies on different mediums. Next, we extracted their DNA and ran PCRs to detect the presence of 16S or 18S ribosomal DNA. Finally, we sequenced the samples with positive results, which allowed us to identify the microorganisms present in ropey maple syrup. In total, we detected 5 different microorganisms, 3 of which are bacteria and 2 of which are yeast. So Leuconostoc mesenteroides has already been detected in ropey maple syrup. It produces dextrins, so it is believed to be one of the culprits responsible for the production of viscosity in ropey maple syrup. In this project, we identified a new culprit that has never been isolated in maple products before, which is Leuconostoc pseudomesenteroides. Finally, we identified the presence of Sphangomonas posimobilis, which hasn't been described as producing dextrins. Next, we identified 2 yeast species in ropey maple syrup, one being a species of Rhodoturula, which has already been isolated in normal maple syrup. We also isolated a species of Cytheromyces. On the other hand, it was never isolated in maple products. This yeast has often been isolated in food products with high osmolarity and is linked to food spoilage. Overall, none of these microorganisms can be directly linked to a human contamination. And in short, this side project allowed us to identify 3 new microorganisms present in ropey maple syrup, including one that is known to produce dextrins. So this concludes the results of our project. And next, I wanted to highlight an important part of our team, education and communication. This is important to us because our consumer survey showed the need to inform people more about science in general. So in the most important collaborative event over the year, us and 3 other iGEM teams worked on the creation of the Rosalind Chronicles. This interactive billboard allowed 26 other iGEM teams to promote and inspiring women in STEM from their country and also promote women in their team. Along with the Rosalind Chronicles, we also collaborated on a workshop titled Women in STEM, Diversity and Inclusion Workshop. We talked about a few important women in STEM throughout history that went under the radar but still had an important influence in their field. And finally, we talked about how iGEM and research teams can be more diverse and inclusive. We also organized an activity called Become a Hero with Biotechnology. In this choose your own adventure format, children aged 8 to 12 design their own microorganism with superpowers such as bioluminescence or quorum sensing in order to move through missions that reflect real problems encountered by scientists. We hope that this will help children gain more knowledge on biotechnology and its potential to help the world. Overall, our team is very proud of the work we have done in communication and education, which goes a lot further than what I presented here. We believe that it is very important for us, for the population to gain more knowledge in science to allow them to better understand what impact science has on their day-to-day lives.
So, to wrap this up, what did our team accomplish regarding the iGEM competition? Well, aside from completing all bronze and silver medal criteria, we succeeded in our proof of concept, education and communication, integrated human practices, excellence in ropey syrup microbiology, and project modeling. We would like to give a special thanks to our sponsors, especially our collaborators de Centre aSAP, de PPAQ, producteurs et productrices acéricoles du Québec, de Québec, and Entreprise Cadorette. Lastly, we would like to thank the members who contributed to the team's wiki, graphic design, communications, and finances.